Okay, so next thing we're going to do is we're going to look at this endomembrane system. This is the second big concept of this chapter. We're going to go over the eukaryotic cell. We're going to go over all the organelles and the job that they have. And then the title here says that they divide labor. So they have very specific things that they have to do. If everybody was doing the same thing, it wouldn't be very efficient. To make it efficient, every organelle has a job to do. For example, mitochondria makes ATP, maybe Golgi apparatus, it's going to package things, it's going to modify and package. Mitochondria, if you look at the size, it's the same size as bacteria. So there's a theory, we're not going to talk about it in this chapter, but later in one of the chapters, that the mitochondria was bacteria and it was its own cell at one point, and then the eukaryotic cells engulf the mitochondria, and in return, basically mitochondria is getting place to live, and then in exchange it's giving it energy. So it was very it makes a lot of energy, it's very efficient. So eukaryotic cells engulfed them, and then they started living in a mutualistic relationship. So we're going to talk about that a little bit later, but that comes from because the size of the mitochondria is small. So this is called endomembrane system, consists of nuclear envelope. This is the nucleus. Around it, you can find the nuclear envelope, endoplasmic reticulum, that's this here. So basically starts here, nucleus, nuclear envelope, endoplasmic reticulum, Golgi apparatus, lysosomes, and then vacuoles, we'll see those in plant cells, and cell membrane. So let's go over this. So this is a photo of eukaryotic cells. So what we're gonna do next, we're gonna do a top view. So next photo is gonna give you a superior view. So this is from top, basically you're looking at, it's a different photo. So I'm gonna go over each one of these and then I'll give you a summary slide. So this is basically still listing. So you have the nucleus here. This is the nuclear envelope, and you can see the spaces in between. Those are called nuclear pores. And this is where the DNA moves through, basically. And outside of the nucleus, you're gonna see the endoplasmic reticulum. And then this is smooth endoplasmic reticulum. There's two kinds, rough and smooth. I'll tell you why each one is called rough and smooth. And then there's ribosomes. The ribosomes are basically making proteins. And then it's gonna move on to Golgi apparatus, that's right here. And it's going to package things and transport them out. In this example, we're using milk proteins. Your book uses milk proteins. So let's look at the nucleus first. Nucleus is going to have the DNA. We're talking about a eukaryotic cell, and eukaryotic cell have DNA inside the nucleus. Nucleus has the DNA, it has the information to make the proteins. So that's the recipe for the proteins. And that DNA is going to move outside. So a messenger RNA, this is a messenger, it's going to carry the information to make the protein from DNA to outside to a ribosome. That's going to move through these pores, nuclear pores. It goes to the ribosomes, and this is where the protein is being made. So this is where the assembly of the proteins is happening. You have the DNA that has the information to make the protein. Messenger RNA is carrying the information outside to the ER, endoplasmic reticulum, to the ribosomes. And that's where the protein is being assembled. The reason it's called the rough ER, because you can see the ribosomes here, and it's giving it a rough kind of appearance. So if you touch it, it's going to be rough. We have some models. It's easier to understand when you look at the models. Whereas smooth, it doesn't have any ribosomes on the outside, gives it a little bit of a smooth appearance here. And after some ribosomes are on the surface of rough ER, so they're on the surface, and then there are some I want to call them vesicles. There are some vesicles that are going to carry these ribosomes to the Golgi apparatus. So there's this vesicle, it's going to carry it to Golgi apparatus. So protein is synthesized, that means it's being made, it's being assembled in the rough ER, then it's carried over to Golgi apparatus with the use of vesicles. Golgi apparatus, it's going to, so what it looks like stack of plates or pancakes, you can see. So it's kind of a stack. This is known as a processing center. It's going to modify and it's going to package. So it's going to do the final check basically. So it's going to modify and it's going to package. Once that's done, it's going to be packaged in another vesicle and it's going to be carried out into the cell membrane. It's going to be carried out. So the proteins leave the Golgi in vesicles that fuse with the cell membrane and they expel the contents outside. So this vesicle is just going to fuse with this membrane and the content's going to go outside. This is the summary slide. This is basically each one of those slides put together. So you start with the nucleus. You have endoplasmic reticulum, and that's where the DNA, nucleus has the DNA that has the information to make proteins. Messenger RNA carries the information to the rough ER, where ribosomes receive the information. They synthesize the proteins, and those proteins are being packaged into these vesicles and sent to Golgi apparatus that modifies and processes it and then packages and then sends them outside through these transport vesicles. So that's whole endomembrane system for you. Another thing I want to tell you, it's either they can go outside if they're ready to go. Otherwise, they might, if there's something wrong with it, I think I might have another slide here. So if there's something wrong with it, then it's not gonna go outside. It's gonna be taken up by the lysosomes. Lysosomes are like garbage disposal. There's this damaged mitochondria. It's gonna take that, it's gonna break it down. So lysosomes, they're like garbage disposals. Vesicles leaving Golgi carry digestive enzymes. So if they're good to go, they go outside. And then otherwise, lysosomes going to break them down if they're damaged. So that's the job of lysosomes. Again, maybe you want to write lysosomes are like garbage disposal. They digest the damaged parts. Here is an animation. So you start with the nucleus. That's where the DNA is. It's going to have the information to make the proteins. And a messenger RNA is gonna bring that to endoplasmic reticulum. It's going to synthesize the proteins, the ribosomes here. Then they are being carried with the transport vesicles to Golgi apparatus. Golgi apparatus is going to modify it and package it. And transport vesicle is going to carry it outside to the cell membrane. And if there is something wrong with it, it goes to the lysosomes and they break it down. So that's the overview of the endomembrane system. And that's a second big concept of this chapter. Are there any questions on this? No, okay. So let's look at some of the membrane organelles that are different in plants compared to animal cells. So most plant cells lack lysosomes, and the cellular digestion in plant cells occurs in vacuole. So you see this big space here, that's a vacuole. This vacuole is going to do the digestion, and it's also going to regulate the water balance. So that's found in plant cells. The green stuff that you see here is chloroplast. That's also found in plant cells. And this liquid part is known as cytosol. That's this liquidy part there. So these big spaces here are vacuoles. They're going to do the cellular digestion, and they're also going to regulate the water balance. And then there are something that are called perioxysomes. Perioxysomes also aid in digestion, but they do that at the ER. So if there's something wrong with the protein that the ER made, these perioxysomes take that and they break it down. So lysosomes, they do that at cell membrane. Perioxysomes do the same thing, same job, but they do it at the ER. So that would be the difference if you were looking for a difference between these two. So let's look at mitochondria. So now I'm showing you some of the organelles that we might not have seen when going over endomembrane system. So we said that mitochondria makes energy. So most eukaryotic cells have mitochondria. It's going to make ATP, and that's what's needed for metabolism. That's what's needed for, everything basically in your body. This is what mitochondria looks like. It's going to have two membranes, outer and inner membrane, and this inner part is called matrix, and then these folds are called cristae. I think next chapter is also going to ask you to memorize these things. So if you want to do it now, or do it next time when we talk about it. And mitochondria is basically going to make energy. And then there's chloroplast. Chloroplast is basically like a solar panel. It's going to take sunlight and it's going to convert it to food. So chloroplasts are found in plant cells or leaves, plant leaves. They are the organelles that use sunlight to produce food for the cell. And the food travels to the mitochondria that extract the energy that's needed for the cellular process. If it's dark or it's at night, it's going to be using that stored energy because there's no more sunlight, so it's going to have to use that stored energy, especially also when it loses the leaves maybe during the winter. It's going to have to use the stored energy, and we'll talk about this a lot more in photosynthesis chapter. Next thing we have is something called cytoskeleton. So cyto, I said it means cell, and then skeleton is basically something that's inside the cell. It's a network of proteins, and it's these three kinds of proteins: microfilaments, intermediate filaments, and microtubules. So these three kinds of proteins are making the skeleton. Skeleton is something that's holding the cell together. These proteins, they not just make the cytoskeleton, they also have a role in muscle movement. They pull and push things. So if you're going to have to take anatomy, they're going to talk about actin molecules, myosin molecules, or tubulin subunits. So they're going to go a lot more in detail for this, but I want you to know that these three types of proteins are making the cytoskeleton. This is what it looks like. It's giving the support and it's holding everything together. I think this is under electron microscope. So this was under electron microscope. It's showing actin, microtubule, and intermediate filaments. I think I have a better picture coming up. So structural support, it's going to give it the shape, structure basically. Another thing, it's going to aid in cell division. When we go over mitosis and meiosis, I said it's going to be pulling things apart or pushing. So these actin molecules pull the cells apart in cell division. Organelle transport. The transport vesicles, they have these microfilaments, they have the cytoskeleton that's basically, that's pushing and pulling, that's how they're moving. So this giving organelle transport. And then cell movement, it's found in the tail of the sperm, which is called flagella, and it's helping that sperm cell move basically. So you're gonna see these three types of cytoskeleton components in sperm tail or in cell division, structural support. So it has different functions. So those are the three I want you to know. Here in this photo, this is a photo from mitosis, I believe. So it's showing that dividing, cell division, microfilaments, actin, and tubulin. And then here you can see it's providing some structural support here, and it's giving it the shape that it has basically. So intermediate filaments, microtubules, and microfilaments, these three kinds. Next thing, I want to talk about microtubules that form cilia and flagella. Let me know in the chat if you heard about these words before, cilia and flagella. Yes. So cilia is more of a brush border or hair-like structures. This is cilia. And then flagella is the tail. It's a sperm cell. You can see the tail. That's the flagella. So cilia is found in your lungs. And basically, when you're breathing, if there is any microorganisms that's going in there, it's getting caught in the cilia. This makes mucus, and it's going to be moved on to your, and you swallow it, and then it's going to go on into your stomach, and that's, you know, where it gets destroyed basically. People who smoke, they basically destroy this cilia. So when they go to sleep and they wake up in the morning, they go in this coughing pit because they destroyed all the cilia. Now the mucus can't be moved. So they get a lot of mucus in the morning and they're going to keep coughing and coughing. So this is what they're destroying when they're smoking. Flagella, it's going to help the cells move. That's for movement. People who have bronchitis, basically they have inflammation of lungs. That might be something, you know, going on with the cilia as well here. This is a flagella, so it's helping the cell move. I think there's a, this, it can be found, it's on sperm cells and it can be found on bacteria cells or other kinds of cells as well. But flagella is the tail basically. In biology, we don't say tail, we say flagella. In anatomy, you're gonna have to learn that the tail of the sperm is called flagella or flagellum. This is showing cilia, hair-like structures. So what this is doing in this photo, it's moving things inside the cell. So maybe there, and it's helping it move as well. So this is amoeba. It's maybe moving some food inside the cell. It's moving water around and helping it move. So these are more hair-like structures, and this is tail-like structure. So flagella and cilia.